Well, welcome everybody to the inaugural Shiley Endowed lecture series that was made possible by the generous support of Darlene and Donald Shiley. And we just want to say a few things at the beginning because it's really important to lay out the partnership that we've had with the Shiley Foundation and how the Shiley Marcos Alzheimer's Disease Research Center at UCSD really was brought to a higher level through the generous support of Darlene. So we're really working today to show how further innovation, collaboration, and groundbreaking thinking within the field of Alzheimer's disease can take us to the next level. And so for more than 3 decades, the Shiley Marcos Alzheimer's Disease Research Center has been leading the charge here at UCSD and nationally. And in 2004, we were even more empowered to extend that work thanks to the generous support of Darlene and Donald Shiley. So just a couple words about Darlene. Darlene is a steadfast, integral, forward-thinking partner for us. She really has touched our entire community. So not only at the Shiley Marcos ADRC has Darlene been making her presence felt, but also throughout our community at San Diego, including the Collaboration for a Cure program that she really spearheaded and has been extending its reach across our local institutes, including the Sanford Burnham Prebys Medical Discovery Institute, the Scripps Research Institute, the Salk Institute for Biological Studies, and the Venter Institute, as well as UC San Diego. So as you can see, we have such a great community here to forward our research against this awful disease and to help the families and the patients themselves so that we can say goodbye to this awful illness. And Darlene really has been at the forefront of that work, and so we're so happy to have her. And so what I want to introduce now is Tom Cerruti, who is the Executive Director of the Shiley Foundation, to introduce Dr. Brad Hyman.
Thank you, Dr. Brewer. Darlene had asked me and Linda Schoaf, another trustee of the Shiley Foundation, to stand in for her. Unfortunately, Darlene has a knee injury, and she'd expected that she'd be able to recover for it, but wasn't able. But she's very much was regretful that she couldn't be here to attend the inauguration of the Shiley Endowed lecture and to introduce such a distinguished first lecture as Dr. Hyman is. The Donald and Darlene Shiley have been at the forefront of philanthropy with respect to Alzheimer's disease research and other neurodegenerative disease research, but also patient care as well. They've been involved in this for well over 20 years, and with respect to this particular institution, probably I'd say around 15 years or so when their involvement with the Shiley Marcos ADRC began. Now the Shiley Marcos Alzheimer's Disease Research Center was named in honor of Donald Shiley, Mrs. Shiley's late husband, and her mother, Dee Marcos, who also suffered from this devastating disease. We are here today. We're very pleased to be here because, again, this lecture series is yet another attempt to move the research forward with respect to Alzheimer's disease. Today, I'm introducing Dr. Brad Hyman, who is an internationally recognized expert in Alzheimer's disease research and is the director of the Massachusetts Alzheimer's Disease Research Center at Mass General Hospital. He's also involved with the Alzheimer's Laboratory at Mass General Institute for Neurodegenerative Diseases, and he is the John B. Penny Jr. Professor of Neurology at Harvard Medical School. In his research efforts, Dr. Hyman explores the neuropathophysiologic and genetic factors that contribute to dementia, and he has authored more than 600 peer-reviewed papers that have been cited in hundreds of publications. He's a member of the National Academy of Medicine, is the recipient of the National Institutes of Health Merit Award, the Potemkin Prize, the Metropolitan Life Award, and the Alzheimer's Association Lifetime Achievement Award. He is integral to some of the most groundbreaking Alzheimer's and dementia research today, and we are looking forward very much to learning about his perspective on what is happening in the field. and how medical and scientific professionals and members of the community can continue to collaborate and for a cure and for new methods of treatment as well, perhaps, and ways in which we can advocate for strong patient and caregiver support systems. Please join me in welcoming Dr. Brad Hyman.
Thank you. Thank you, Tom. Thank you, Linda. And if you'd pass on my thanks to Mrs. Shiley as well, please. It's really an honor and a privilege to be here and to help kick off the annual Endowed lecture here. I have many happy memories of UCSD. I view Bob Katzman and Bob Terry from years ago as being the heroes of science, and Leon Thal, of course, being a just dominant figure in the field and a close friend as well. And so for many years, I've should speak more loudly about how wonderfully I have always thought of them. What I'd like to do today is share some of the recent work that we've been doing, thinking about Alzheimer's disease as a process that takes years and years and years to play out. And it's one of those things which is really sort of difficult to think about in the laboratory. In the laboratory, we're very good at doing experiments that start in the morning and hopefully end by the mid-afternoon. Certainly, you want a series of experiments that don't last any longer than a what we call a postdoc life, which is 2 or 3 years of training for a student or a postdoc. And yet Alzheimer's disease doesn't obey any of these rules. It really starts out maybe a decade or so before patients show up in the clinic. Takes another year or two before a diagnosis is really established, then another 8 or 10 years as the disease plays out. And so how do you model a disease process that stretches out over the course of maybe 20 years, half of which is clinically silent, in a way that helps you figure out how to intervene in that process and slow it down or stop it? And so we've been struggling with this and trying to think about different ways of examining the disease process at different stages in sort of a cross-sectional way, so that we can go, well, in a year it looks like this, in 5 years it looks like this, and 10 years it looks like this, and then take those snapshots and kind of put them together into what might be a kind of a movie of what all those pictures would look like. There's some problems in doing that, of course, and I'm reminded of a colleague of mine who pointed out that if you did the same thing for a baseball game, a Red Sox game in particular, what you'd find out is that there's a lot of people standing around drinking beer. If you just took one picture every hour or so during the course of the 4 or 5 hour Red Sox-Yankees game. And while that might, in fact, be the point of baseball, still, there's something more to the game, which is, of course, that the Red Sox end up figuring out a way to lose at the end, no matter what's happened in between. So what we've been trying to do is figure out ways to model this process so that we can do more than just get snapshots. And I'm going to be presenting really 3 stories. And they have to do with this whole question of how the changes that we see in the brain at autopsy relate to the kinds of clinical symptoms that occur in Alzheimer's disease, and what are the molecular and biological mechanisms that lead to progression of the disease. So I'll be focusing primarily on neurofibrillary tangles, which are these tau-containing lesions within a specific set of neurons in the brain. I'll mention these amyloid plaques, which are distributed throughout the brain and occur very early on in the disease. And of course, these lesions are associated with neuronal death and loss of neural systems. And it is that loss of neural system integrity that ultimately leads to the symptoms of Alzheimer's disease. Part of the question is how Alzheimer's disease gets worse over time. So people don't wake up one morning and are demented, with the possible exception of undergraduate students on Monday mornings. Aside from that, though, it's this gradual process which I've described. And if you look neuropathologically at cross-sectionally individuals who have died at various points, presumably in the disease, there's a very consistent hierarchical pathway that seems to be affected, with those areas of the brain that are most involved in memory formation, the limbic areas, entorhinal cortex, hippocampus, and so forth, affected first, and then the disease seems to spread out from those areas, and later on, other areas of the brain that have to do with language and judgment and so forth and involved. And ultimately, the entire cortical mantle is involved in Alzheimer's disease. And this has been given a nomenclature from Heiko Brack and called the Brack staging. It goes from stage 1 to stage 6 of early to late. And it seems as if there's this progression of disease that matches pretty well, at least in general terms, with the progression of clinical symptoms from an early short-term memory impairment to more severe memory involvement and some additional language difficulties and so forth. And so in general, it seems like this progression of disease matches with where tangles form, and also where neuronal loss is, because those two things really do match very closely to one another. Even within these big brain areas like the limbic system, the neurofibrillary tangles occur in very unique populations of cells. And so in the entorhinal cortex, which is the primary area which helps with encoding of new memories, it's layer 2 of the 6 layers of entorhinal cortex, specifically that's affected by tangles. And interestingly, those are the cells that anatomically give rise to the projection to the hippocampus. So those are the cells that provide information to the rest of the brain as to what one is supposed to be remembering. And early on in the disease, those cells are affected and then affect the hippocampus. So one of the questions that's puzzled us over the years is, how is it that the disease spreads from one area to the next? And then also, what's unique about those cells that are particularly affected? There were two ideas that we puzzled over. One is that the cells that are affected tend to be very similar to one another. They tend to be large, they tend to be glutamatergic, they tend to have a major projection with relatively sparse projections. They tend to have other biochemical characteristics. Maybe they're just vulnerable, and it turns out that everybody who's blonde and blue-eyed is more likely to get a sunburn, and so there's lots of people who are blonde and blue-eyed, and they all get sunburns, and some people are a little more sensitive than others, but fine. That it's something about the fundamental characteristics of the cell which makes them vulnerable to get tangles and ultimately die. The other possibility, and something that we thought about early on, was that the neurons that were affected were not universally, but by and large, connected to one another. And was there something about the fact that they were connected that led to their vulnerability? And I'll fast forward to the current thought, which is that both of those things is true, that there's both something about that's unique that makes some types of classes of neurons vulnerable, and also the fact that they're connected to one another seems to make them vulnerable. So the 3 questions that I'd like to pose in the course of the next 30 or 40 minutes is, does tau propagate? That is, do these neurofibrillary tangles actually go from one cell to the next cell? Does the fact that the cells are connected, is that why they both can develop these tangles? And I'll emphasize tau, of course, is the major molecule that makes up these tangles, and so I'm using them somewhat interchangeably, although in fact, in many of the experimental models, I'm going to be using a mutant form of tau, P301L, which actually clinically is associated with frontotemporal dementia. And I just want you to forgive me that little interchange of work. The second question is, does amyloid impact tau propagation? I mentioned that tangles occur in these very hierarchical distribution, one area of the brain, then the next, whereas the amyloid deposits, and amyloid, which has been the real key in terms of therapeutic interventions for the last decade at least in terms of pharmacologic development. Amyloid tends to occur throughout the cortex, tends really not to be associated with clinical symptoms in any way that we can measure. So there are people with a lot of amyloid who have no symptoms, people with hardly any amyloid who have more symptoms. It's not that closely correlated. And the places in the brain that are affected by amyloid aren't really the places in the brain that give symptoms. But nonetheless, everyone who has Alzheimer's disease, by definition, has a lot of amyloid in their brain. And so one of the puzzles we had was, well, most diseases only have one lesion. Well, why does Alzheimer's disease have two lesions? And is there something unique and synergistic about that fact? And then the third is some new data in which we're curious to understand how it is that the soluble tau, which is a normal molecule in every neuron, somehow decides to aggregate and condense and form these neurofibrillary tangles, which seem to be the critical toxic bioactive molecule. And with the exception of this sort of introductory piece, all the data I'm going to be showing you is not only not yet published, but much of it is fairly early days, and so not all the I's are dotted and all the T's are crossed, but this is such a rich community of neuroscience. I very much wanted to share with you what our current thinking was. So first the question, does tau spread, or is this underlying vulnerability in susceptible neurons? In order to address this question, we built a mouse model, actually using Mark Mayford's driver line, because I saw the windows from his lab as we walked over here this morning. So Mark, I don't know if you're here, but thank you. And this was a line that makes tau only in the entorhinal cortex, that area of the brain which is primarily the first place that's involved in Alzheimer's disease. When I say tau is driven only in the entorhinal cortex, I mean only in quotes. It's a mouse after all, it doesn't really know. It's mostly in the entorhinal cortex, and for the purposes of the work shown here, that's sufficient. And you can see here in this orange stain where the tau is in entorhinal cortex here, in pre and paracebiculum. And then this line here are the terminals of these cells. So these cells project via the perforant pathway to the middle portion of the molecular layer of the dentate gyrus. And so you can see the end points, the terminals of these cells right in that line. And so this is a relatively young mouse. It's doing just what we expected it to do. And as the mouse ages, something really unexpected happened. So as the mouse got to be 12 months of age and then 18 months of age, and then, well, I can tell you what happened at 18 months of age. The graduate student who was working on this came into my office and said, "Am I ever going to graduate?" And I said, just wait a little while longer. And so at 24 months of age, something astonishing happened. And that is that the cells that the entorhinal cortex projects to, which are the dentate gyrus granule cells, developed human tau. And surprisingly, there was no messenger RNA in the dentate gyrus granule cells. So the only way that this human tau could have gotten there is if it had been released from the entorhinal cortex and then taken up by the dentate gyrus granule cells. This was a little controversial when we first proposed this. We went so far as to do laser capture, pulled out the individual cells that had the human tau immunostaining, did qPCR to detect as few as 10 copies of messenger RNA for human tau. There wasn't any there. And we did a variety of other sort of control experiments and came to the conclusion that this really was a feature of the mouse model, that tau could be released from one cell, taken up from the next cells. We could find human tau in rare microglia and astrocytes, again suggesting that it was released and taken up. And then at essentially the exact same minute that we published this paper, Karen Duff's laboratory published the exact same observations in another line. And so it became not just a crazy thing that Brad was thinking, but a crazy thing that Brad and Karen were both thinking. And that, I think, really helped make us a little bit more serious about whether or not this could be true. So the first couple of years after that observation, which was in 2012, was really trying to convince ourselves that tau really did propagate between neurons, because tau is an intracellular protein. It's not supposed to get out of the cell. It certainly shouldn't be taken up by the next cell. There's certainly no reason in the world to think that any neuron would take up a misfolded protein and let it hang out there for a while. And it had a variety of implications, which have been broadly called prionlike propagation, which a term that I don't particularly like in terms of its implications for prion biology, but which has a thought about changing the protein conformation based upon a misfolded protein that instructs the protein in the next cell for what goes on. So we wanted to understand really the underlying biology. Was this true, or was this just something that happened in a mouse? And we wanted to understand then each one of those processes that I just mentioned. So if this whole process is true, you'd expect that there would be tau in the cell body. It would go down the axon. It would accumulate at the synapse. It would be in both the pre and postsynaptic sites. It would then be able to find tau in the next cell and instruct that tau to misfold and so forth. And each one of these steps would have a specific kinetic process associated with it. And so we've spent some years developing the tools to try to measure the kinetics of each one of these steps. And I'll just jump to, again, conclusion. It turns out that each of these predictions, that tau is released to the synapse, that tau can be found at the synapse by EM or by some other techniques that we developed, that we can stimulate tau to be released, they can be taken up by neurons. All of that can be demonstrated experimentally in a variety of systems using both mouse models and now in more recent work in human as well. And we've even gone to the point where we have a pretty good handle on what specific form of tau it is that has these biological properties. So to begin to explore this, I'm going to show you data both from the human and also from the mouse. The mouse model we use is the mouse 4510 transgenic model that we developed with Karen Ash some years ago. It has the advantage that it is a doxed response element driving it, and so you can turn it on and turn it off. And essentially everything that I'm going to show you from the human and from the 4510 mouse are interchangeable. The mouse overexpresses human tau by 10-fold, and so for some experiments the mouse is a little more convenient to use, but everything that we've done so far really works in both systems. So in this first experiment, we took soluble tau from a human Alzheimer brain or from a control brain that didn't have Alzheimer's disease, and spun it down and at either 3,000 G, which is enough to sort of pull down nuclei and cells and debris, or 150,000 G, which is enough to pull down molecular large molecules. And then we took the supernatant and added it to neurons in culture. And the 3,000 G spin, but not the 150,000 G spin, was able to be taken up by either by mouse neurons and be detected by an antibody that specifically saw human tau. And so we had an assay in which we could begin to sort out what kinds of tau was able to be taken up by neurons. And we could demonstrate that depending on how we did the fractionation, we could see the tau either being taken up or not taken up. So we had an assay to read it out. Next question was whether or not once it's taken up, does it hang out there? Does it rapidly degrade? Does it go down the axon? Can it be released? To do those experiments, we used these 3-chamber microfluidic devices that Shuko Takeda in the lab designed. In these, the neurons are plated into a small plastic dish, which allows the axon to go down a small channel, interact with other neurons. Those neurons send their axon down the next channel, interact with further neurons. You can sort of do this on and on and on. You have to be really careful with how you plate them to get the axons to all go in one direction. But it turned out that Shuko very cleverly discovered that gravity was an aid, and if you simply set the wells upright when the neurons were plating, you could get the axons to grow in the right direction. And so we have this device. If we add the high molecular weight tau to the first chamber, it gets transported down the axon, released, taken up by the next neuron, transported down the axon, released, and so forth. And so this idea of propagation, of having a specific form of tau taken up, transported, released, seemed to be true in experimental models. We wanted to understand more about this, and so we put it over a size exclusion column, which is a very mild extraction technique. This size exclusion column has big stuff that comes out early, little stuff that comes out late, and it's marked here as high molecular weight tau, and the monomer dimer tau, the big peak. This is from the 4510 mice. The equivalent figure from the Alzheimer brain shows that the high molecular weight tau peak is really, really, really small. And so in an Alzheimer brain, about 1% of all the tau is present in this high molecular weight fraction. The rest of it is monomeric. This is out of the soluble fraction of tau, so this is not using neurofibrillary tangles, but just what comes out in a TBS extract. But in every instance, it's only the tau that comes down in the high molecular weight fraction, which we estimate to be a couple 100,000 molecular weight, which is bioactive and competent to be taken up by neurons. And so in the lower panels, you can see the green is the human tau. Only those early fractions are taken up, even though there's 99 times more tau in the low molecular weight fraction, it's not taken up. So again, this gives us an assay and a way to identify what's unique about this tau species. And just to take that story a little bit further, we've gone on to use immunodepletion assays to characterize that tau. The far C terminus is missing. There's a variety of phosphorylation sites that appear to be differentially present in the high molecular weight species, and there's some conformation changes, and we just are now doing some mass spec experiments to really try to define what that unique bioactive species is. But it's the sort of critical thing is that it's a rare species, meaning roughly 1% of all the tau that's present, which of course is just Mom Nature's way of pulling our leg and saying it couldn't possibly be easy to figure out what this stuff is. We still wanted to convince ourselves that this notion of propagation was true, and we wanted to develop other model systems to begin to examine whether or not this was true in an intact brain rather than simply in cells and culture, because cells and culture really aren't exactly the same as the intact brain. And so I just present two bits of data here. One is if we inject this high molecular weight species or the low molecular weight species into an intact wild type or P301S overexpressing mouse, it turns out that in the intact brain, neurons take up the unique bioactive species. They don't take up the low molecular weight species. And it actually hangs out there for at least a month, which is really astonishingly long time. So it's a very stable conformation. On the right hand is a panel in which we developed an AAV system in order to ask the same question. This uses a green fluorescent protein and an iris-like device, which is a 2A, and then the human tau. In this case, it's P301L, although we've done this with wild-type MAO and with various other mutations. You inject it into an area in the brain, shown here, the entorhinal cortex, and if you wait 8 to 10 weeks, you can find human tau positive cells that don't contain the green fluorescent protein in the projection zones of the entorhinal cortex. And so instead of having to wait 24 months, which my poor graduate student Alex de Calignon had to do for that first experiment I showed you, now we're able to do experiments in 8 to 10 weeks. And interestingly, we're able to now do experiments in mice that are different ages to start with, or in mice that have different genetic backgrounds, or mice that overexpress various things that may accelerate or protect against this phenomena. And those experiments are underway, but I'll share with you at least some preliminary data that I just saw the first blush of last week, which is that if you do this injection into old mice and you wait 8 to 10 weeks, you get about 4-fold more propagation than if you inject into young mice. And so that whole puzzle about why it is that aging is an important part of people getting Alzheimer's disease, we feel like we're beginning to try to get a little handle From a biochemical perspective, this high molecular weight species is shown here on an SDG age plot. The 4510 mouse creates a form of tau that propagates. The 21221 mouse has a different form of tau. This is a wild type tau that's slightly lower expression levels. It doesn't generate this high molecular weight smear. It also doesn't propagate. And so again, we have a built-in sort of experimental control for the mouse work. And here again, we're able to inject this into the mice, or we're able to show that if we use a reporter, which is the tau-RD FRET-based reporter, that this high molecular weight species can be isolated, put onto cells, and form aggregates. So all of this is exciting and wonderful and actually quite a bit of fun because the experiments work. But really, it's all based on postmortem samples. It's all based on mice. It's all based on a variety of systems, which if one wanted to be self-critical, which of course in Boston we would never be self-critical, but if one wanted to be self-critical, you'd say, well, gee, that could all still be an artifact. Prove to me that this really is relevant to patients with Alzheimer's disease, and not just to what happens in tissue, which has already died. And so in a heroic set of experiments, Shuko Takeda took 20 patients with Alzheimer's disease spinal fluid, 20 controls. Half of those were from our own CSF bank at Mass General. Half of them were from the University of Washington. Elaine Peskin and Tom Montine were kind enough to work with us on that. And we wanted to make sure that anything we found was true and from 2 different sources of samples and 2 different freezers and 2 different kinds of plastic tubes and 2 different people who did LPs with different kind of needles, and that really it was going to be robust, that it was sort of true, at least as best as we could find. And we went searching for this high molecular weight species in the spinal fluid. Now, I've emphasized to you that this high molecular weight species is about 1% of the total soluble tau. And since tau in spinal fluid is detectable, but not trivially so, to start with, if you're trying to find 1% of that, it becomes difficult. And Shuko had to develop a variety of biochemical methods to start with a lot of CSF and really nail it down. And this paper that we just published in Annals of Neurology demonstrate that in Alzheimer patients, but not in controls, and interestingly, not in frontotemporal dementia patients, that we can detect this high molecular weight tau, and that the amount of the high molecular weight tau was sufficient in at least 2 of the instances where we had enough spinal fluid to start with to isolate it, that we could demonstrate that the high molecular weight tau that came out of the patients, out of a living patient with a clinical diagnosis of Alzheimer's disease, was sufficient to propagate in the experimental models that we had used. And so in summary, it seems as if we've isolated a form of tau which is sufficient to propagate, which appears to be responsible, at least to some extent, for tau moving from one neuron to the next neuron, that it can be found in minute quantities in spinal fluid from Alzheimer patients, that even that tau can be released and taken up and transported down axons and propagate. And that we've developed a variety of experimental systems, and part of what I wanted to do in this lecture was to describe the experimental systems, because with any luck, they'll be of some use to other people as well.
Is this an aggregate then, or is this
It's not a perihelical filament. It's not a beta pleated sheet. It doesn't come down at 3000 G. It doesn't, by EM, it's amorphous. So, nor is it monomeric. So, our current guess is 3 to 6, but we could be wrong. All right. I mentioned that amyloid in the brain is a consistent feature of Alzheimer's disease. And there's this interesting neuropathologic, relatively rare condition in which people get tangles but without any plaques. And it's called PART, P-A-R-T. And in that instance, the tangles almost always stay confined to a BROC1-2-ish place. So mostly in the limbic system, but hardly ever get out to the cortex. Whereas in patients who the much more common instance of having amyloid in the cortex, the tangles, of course, get out there too. And the tangles in part and the tangles in Alzheimer's disease are indistinguishable, at least to everything we've done. So you immunostain them, you look at them, you scratch them, you tell them jokes. They all respond just exactly the same way. We wanted to know, however, whether or not there was something uniquely different about the tau in the brains of patients who had amyloid compared to the tau in the brains of people who didn't have amyloid. And there was a variety of work in the experimental literature, and I'll show you some from our own work as well, that suggested that there's some synergy between tau deposition and amyloid. Here I just show that the symptoms of Alzheimer's disease really begin when those tangles get to the cortex. And so from the perspective of this sort of clinical pathological correlation, figuring out what it is that trips that trigger, that gets tangles from more or less confined to spread out over the brain, would potentially have a clinical impact. So when we take the mouse that only has tau in the entorhinal cortex, and we cross it with a mouse that expresses APP-PS1, this is the Jack's mouse, and so it has amyloid throughout the cortex. And we do that same experiment. Remember, it took 18 to 24 months to see tangles in the dentate gyrus in the entorhinal cortex only mouse. When we cross in the amyloid, so we reproduce something that looks sort of more like Alzheimer's disease, that takes 10 to 12 months. And the number of neurons that develop tangles increases by 2 orders of magnitude, and the amount of neuronal loss, again, increases by 2 orders of magnitude at a much, much earlier phase. So there's something about amyloid being present in the cortex that accelerates this propagation phenomena, which matches up really well with the clinical data that we have. And so this just demonstrates that the number of neurons that remain at an early time point, this is at 10 months of age, in the initial entorhinal cortex only mouse that's just normalized to be 100. When you haven't changed the tau at all, and in fact the amount of tau hasn't changed, but you've added the plaques, you see that the number of neurons that remain goes down by 90%. So in terms of neurotoxicity, which is what I believe is the final common denominator of the neural system failure that drives the symptoms of Alzheimer's disease, the presence of plaques, even though there's very few plaques in the entorhinal cortex in these mice, the presence of plaques throughout the cortex sets up a tissue-specific response, which drives neuronal death. And not surprisingly at this point, when we do the biochemistry on this, all of this correlates with this high molecular weight smear, which is hyperphosphorylated, shows up on the SEC column in those fractions 2 and 3, and so forth and so on. When we use the seeding assay, which is the Fred assay that I mentioned before, and we asked whether or not the tau in those APP crossed mice is different from the tau in the mouse alone. And again, the tau driver is exactly the same in both of these. It turns out that using the seeding assay, the APP-PS1 crossed 4510 mice have roughly 3 to 4-fold increased amount of seeding for exactly the same amount of input tau, so that the tau is much more bioactive when amyloid is present. Now, amyloid by itself leads to no seeding, and in fact, we can immunodeplete the mixture that comes out of the APP-PS1 4510 cross for A beta, and the immunodepletion doesn't affect the seeding. So there's something about the conformation of tau which has changed, and it looks as if more of it has converted into this high molecular weight, hyperphosphorylated form. And so we begin to have now a biochemical handle, a molecular handle, on what it is that changes tau to make it more bioactive, more likely to spread, more likely to lead to potential neurotoxicity. Again, all this is in the mouse. And so we came back to that initial observation of part versus the sort of equivalent of PART, but with plaques as well. We had half a dozen cases of each in the brain bank. We walked down the hall, and Matt Frosch found them. We selected out various parts of the brain. And of course, when the human material, you don't have all the controls that you have in the mouse. But bottom line is that the same phenomenon occurred. And you can focus on the lower right panel there, where the red bars show that especially in areas where there are terminals, the brains that had amyloid have, again, roughly 2 to 3 times more propagation than the brains that didn't have amyloid, despite having been normalized for the amount of input tau to the assay. So that there is something about the presence of the amyloid in the cortex. And that doesn't have to be amyloid. It could be microglial activation. It could be astrocytic activation. It could be, many, many things. But there's something about that combination of amyloid and tau in the cortex that converts tau to a different molecular species, which has the properties that it allows uptake, propagation, and instructed templating of tau in either mouse or in some recent work in iPS cells in human tau as well. So I just want to spend the last 8 minutes or so talking about another transition. The focus, as you can gather from this first part, is that soluble tau, rather than the filamentous neurofibrillary tangle, seems to be the bioactive species. And this is a little disappointing for somebody who trained as a neuroanatomist and as a neuropathologist, because I never could quite and because you look under the microscope, and it must be true what you see under the microscope. And you can see tangles, whereas you can't see any of these soluble things. Those are all sort of figments of a biochemist's imagination. And so it's a little disappointing to learn that really those things that Alzheimer himself saw and that earned the name of the disease after him really may not be the relevant bioactive species. But still, obviously they're involved in some way. And so how can we relate this soluble oligomeric hyperphosphorylated form of tau to the neurofibrillary tangles? And this is a challenge that Sue Zwegman and Bahar Efkabar took on. And this is very preliminary data that I just want to share with you, because it brings a different kind of biology to bear, which is pretty exciting. Now, tau has always been viewed as being an intrinsically disordered protein. So most proteins, of course, have a very standard structure. They come off the ribosome, they fold, and they do whatever they're supposed to do. It turns out that tau doesn't really do that. By NMR and CD physical biochemistry approaches, tau flops around. And it's only when it's associated with microtubules does it adopt a formal structure. And even then, the N-terminus sort of forms this sort of fuzzy coat around the microtubules. By atomic force microscopy, which we did with Sue Zwegman a couple of years ago. And so in the last 2 or 3 years, several groups highlighted by Tony Hyman, who has no relationship to me at all. And yet I desperately want to publish a paper with him. And actually we have one submitted now from this work. Tony Hyman described in the setting of ALS-associated molecules a funny property of intrinsically disordered molecules in which they can sort of glum together and they form a liquid-liquid phase separation. And it's a little bit like oil and water, that if you, but make that in your head a little bit more miscible than oil and water is. So more like glycerol and water, that under some circumstances you can get little droplets of glycerol, but if you shake it up, it completely goes away. Now make believe that that's completely reversible and under the cell's control and can happen in seconds, and you have Tony Hyman's notion of phase separation. And on the surface of these little droplets, of liquid droplets that occur within cells, catalytically important reactions occur. And in the instance of some of the things that Tony has demonstrated, they can actually take on some of the functions of immediate response genes to stress. And so there's really interesting biology about what's happening. And essentially, it's a way for the cell to immediately and reversibly create something that looks like an organelle. So this was true for a small handful of proteins that Tony had explored. It turned out that tau really didn't follow the rules that Tony had laid out for what kinds of proteins would be intrinsically disordered and be able to form these liquid-liquid phase separations. But in some ways, when we considered what might happen to tau, it conceivably could form these LLPSs. And so Susan Bahar went down to St. Jude's and in that laboratory did the first set of experiments asking whether or not tau might form these this LLPS, the liquid-liquid phase separation droplets. And of course, I'm telling you this story, so it turned out that it did. And not only does tau form these droplets, but it turns out that the high molecular weight, hyperphosphorylated tau forms them beautifully. Recombinant tau, low molecular weight tau doesn't. And intriguingly, mutant tau, which causes frontotemporal dementia, that P301L mutation, for example, that I suggested to you, does without the benefit of phosphorylation. And if you block phosphorylation in any of a variety of ways, if you use phosphatases and get rid of the phosphates, if you take a recombinant tau but introduce artificial sort of pseudophosphocytes along the way, it does. And so it really, it's completely true with isolated protein on a slide under a phase contrast microscope. You can watch them happen. It happens in a few seconds. They're really cool. They form these little droplets. I actually had a movie which wouldn't play on my Mac, and so I didn't bring it in. But you can just kind of watch them swirling around and wetting surfaces and doing just what's supposed to happen. Yet we challenge that this is a cool system in vitro, but does it happen in cells? And does it happen in neurons in vivo? And does it happen in the Alzheimer brain? And suffice to say that all that is true, and that also, although they happen very rapidly, they also sort of mature, and if you leave them sit around for 5 to 10 days, they become thioflavin S positive. So they don't, but the EM doesn't look like a PHF. That would be way too nice. But there's some sort of maturation, which we think is probably an oxidation phenomena, in which these LLPSs actually sort of mature. So we think that they may be a way between this notion of a high molecular weight species, which has certain biophysical properties, and ultimately the development of this aggregate of tau in the cell body. So we're not sure what the trigger that changes these high molecular weight soluble, hyperactive charged molecules is that forms LLPS. We don't know exactly whether these foci form. We can actually watch them form and dissolve in cells. We now have a way to do that with multiphoton microscopy, so that we can do that in the brain of a living mouse. These beta-pleated sheets form relatively quickly in the course of a few days. That was especially satisfying to me because some years ago we published a paper in which we'd done multiphoton microscopy of transgenic mice that overexpressed tau and formed tangles, and we were able to observe tangle formation in the course of a few days, and that always seemed like an odd thing, that was just a bizarre thing for a mouse. And even though nature was kind enough to accept the paper, it still seemed like a bizarre thing. And so, to see that same sort of biophysical process actually happening in under controlled conditions also seems like it might be true. And then maybe the most intriguing thing to me about this whole new story is that FUS, TDP43, HRNP, so ALS-associated proteins all have the same biophysical property. And that's where J. Paul Taylor, who was down at St. Jude's, where we did the initial experiments, and now more recently Tony Hyman as well, have been focusing their energies, because these are proteins that undergo this same biophysical change under similar conditions, under phosphorylation or in mutant forms that are associated with these diseases. And so it feels as if this is a biophysical property that can underlie ALS, frontotemporal dementia, and Alzheimer's disease. And if that's the case, we might be getting closer to the truth of how it is that neurodegeneration leads to neuronal system dysfunction and death. So I'll just finish there, quick summary here, and then thank especially Suze and Bahar, who did this most recent work that I mentioned to you, acknowledged Karen Ash and George Carlston, with whom we did all the animal modeling, and of course thank our generous folks who provided the support for this work. So thank you very much. Well, so until this recent LLPS work, the answer to why tau would form these aggregates as opposed to some other protein was just that was a mystery of mom nature. And now it seems as if, at least we're playing with the hypothesis that these intrinsically disordered proteins, and it turns out that it's about 2% of the expressed genome fall in this category, at least by, depending on where you put your cuts. And so it's given us some targets to look at to see if any of those other things glom on with tau and form it. But at least it might explain why tau can adopt these unusual structures that are especially stable. And it may well be that unless they're stable structures, that they don't accumulate in the brain, and the neuropathologist never sees them. So exosomes would be membrane bound, of course, and these are not membrane bound by multiple different markers. They also have a different viscosity, so we were able to find a fluorescent dye that was actually used in sort of the polymer physics world, and apparently is a useful thing if you're doing microelectronics for some reason. But anyway, it's this fluorescent dye that changes its fluorescent properties based upon the viscosity of the solution it finds itself in. And so we're able to actually label these with the viscosity dye. And then when we change the conditions so that they dissolve, it goes away.
My second question is, what happens in the presence of beta?
Great question. Our initial experiments, not surprisingly, that was one of the first things we did to cells. Nothing happens in the presence of A-beta as far as we can tell right now. So, we'll keep tweaking with this, but that was a low-hanging fruit that would have been just wonderful, but data are data. Stuart?
Right. Well, that was an amazing. So, beside the exosome paper, which is I think the Intrinsic paper, the other major point of that paper was the depletion of microglia aggregated to the population. So whether it's the exosomes in the form of microglia, lipids, so have you thought about that experiment, done that experiment by depleting microglia?
So we haven't done that. In terms of the propagation between cells, we can block that with really to 90+% with antibodies. And so in my mind, that argues a little bit against exosomes, because you'd expect that the tau would be protected from access from antibodies. And you wouldn't think that tau would be on the surface of the exosomes, and demonstrably isn't, as it turns out, because we can isolate exosomes and look. So at least in the model systems we have, it doesn't appear as if the exosomes are playing the major role. But I don't want to. It's it's sort of a negative result, and I don't think that you can. It would be hazardous to overinterpret that. I'm sorry. I'm a deep believer that we don't really understand what microglia do, and if there was a role for microglia, I'd be happy to accept that. I don't know that. Again, these are all model systems, and you want to be thoughtful about what's specific to the system and what sort of real biology that's relevant to the disease. So that's a great question, and we had thought that it was just sort of stochastic, that it was a rare event, and that you just had to wait a long time for it to eventually happen because it was a rare event, and sort of like the Red Sox winning the World Series. If you waited another 100 years, it might happen again. But our recent experience with the AAV2A construct suggests that there is something fundamental about aging that either is changing the recipient cell or the release. It doesn't appear to be the release in early experiments, and we don't have the biochemistry back yet to know whether or not it's fundamentally changing the physical properties of the tau that's being released. So that's a great question, and I hope we'll answer it soon. Or at least one very, very, very hardworking postdoc. Yeah, no. So don't have any answers. Of course, the whole issue of what happens in aging to make people vulnerable for all of these diseases is uncertain. If in fact it's this, if you take this last notion of the LPS and ask what it is that stabilizes it and keeps it around, and so that it matures and aggregates and then doesn't dissociate, the viscosity actually increases over that time. That's the sort of process that I could imagine would have altered with aging, in which any number of stresses like oxidative stress and so forth that might be associated with aging in general, and that it's all sort of a kinetic stochastic property, that it's just more likely to hang out for a longer time in an old brain than in a younger brain. So that's our working hypothesis, but it's not. Doug.
Brad, thanks very much for the briefing talk. So, on whether these aggregatable forms, and whatever the best word is, these high-molecular forms of tau, how are they handled by cells? Can they spontaneously disaggregate? Can they be degraded? Or if they hang out long enough, is it inevitable that something's going to go wrong?
So again, thinking about this in terms of kinetic processes, if you seed cells that have a bioreporter in them that's fluorescent, you can watch over the course of the first 3 or 4 hours the seeds form, then they grow up, and then they stabilize. And if you continue watching them for 36 hours, about 10% of the cells that have seeds in them, you can't detect the seed anymore. You can see the seed, and then over the course of the 36-hour time period, it no longer is present, even though the cell is still there, which might mean that our optics are lousy and we lose 10% of them, or it might mean that the cell actually has the capability of degrading them to some extent. I favor the latter interpretation, not because I have so much faith in our optics, but because it feels like the biology ought to have a forward reaction and a back reaction. But that's literally last week's data. So, but we're trying to develop the tools to get at that, because wouldn't it be cool if we could figure out a way to accelerate that back reaction?
Putting together all these comments and a potential explanation of the aging component, have you started looking into the clearance mechanisms? Because when we were working with alpha-synuclein propagation, what we observed is, and you mentioned something, right? It's not the donor, it might be the recipient cell. And working with knockout backgrounds that don't express endogenous alpha-synuclein, we see that stops propagation, and in fact, when you peel down in very short times, propagation keeps happening, but those cells that are kind of orphan or the autophagy system regularly able, they just clear it so fast that we can see the accumulation. So coming back to this interaction of amyloid and tau, we might think that you have already a load of amyloid that is blocking clearance, and now tau is being pulled and sustaining those cells.
Yeah, that's exactly the sort of notion that we favor, which is that we've done the crosses with tau nulls, for example, and again see propagation continue to occur, but it doesn't stabilize because the endogenous tau isn't there to misfold and sort of reprime the pump. And it's entirely plausible that the presence of A beta is doing something like simply causing the proteasome to be overwhelmed and therefore allowing the tau to accumulate. So that's what our thinking is. Yeah.
So the propagation that you apply those through a synaptic mechanism implies that there may be a very specific cell biology, subcellular biology to this if they're propagating through synapses rather than through adjacent cells.
So I should be careful. We've demonstrated that it can happen transsynaptically. I'm pretty convinced it also happens locally. And so some of the systems that we've developed, like these microfluidic devices, are designed to highlight the propagation across synapses. But I'm not positive that that's the only place it happens, and I'd be surprised if it were. I'm sorry?
It may not require a synapse.
Yeah, I don't think it does. And in fact, embarrassingly, you can use hex cells for all these assays too. And so kidney cells don't.
It was about, you looked at the connection to ApoE biology.
You'd think I would have. Not yet.
It's all very exciting.
Thank you. Okay, well, we're a little late. I don't want to keep people from their appointed rounds, but
Reception, so thanks very much for having us.
thank you.